Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 194th seminar, and I call it a University of Houston Day. Finally, it's April, and as T.S. Eliot, the poet, said, in the wasteland, April is the cruelest month, breeding lilacs out of the dead land, which I saw at Botanical Garden in March, a bit earlier than usual. I saw huge temperature swings within a day from early 20 Fahrenheit to mid 80s or 30 Fahrenheit to 90 Fahrenheit in St. Louis. Climate crisis is real, and I believe we should solve this global problem by international collaboration. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Patrick Cirino. He is a professor at the Chemical and Biomolecular Engineering Department of University of Houston. I met him for the first time roughly 2 decades ago when I was presenting a poster as a graduate student, and also when he was evaluating my poster as a poster judge. I don't believe he remembered me, but I met him and clearly remember him. At the time, I was working with Christopher Prader, who is now at MIT Chemical Engineering Department head. And then by joining the Chris Voigt lab for my postdoctoral research, Patrick became my academic uncle because Chris and he worked with Professor Francis Arnold, a Nobel laureate in chemistry and the first speaker of this webinar series in 2021. So Patrick is, of course, as you know, a leader in protein engineering and biocatalysis, and I'm so glad finally to host my academic uncle today. Patrick, thanks so much for your contribution to the scientific community, and the virtual podium is all yours now, and thank you again.
Thank you so much for the intro. I don't know if I remember that poster session, but I feel like I've known you for 20 years. So I really appreciate your inviting me to participate here today. And, you know, Chris Voigt was actually also my roommate for quite a while. So I don't know if that probably just still makes it uncle, but all right, let me share my screen. Do you see it?
I see the slide mode, please. There we go.
Good.
Go ahead.
You go back?
Oh, no, please go ahead. Yeah.
Oh, okay.
Perfect.
Sure.
Thank
okay.
You.
Sure. Okay. So yeah, I'm going to start my little timer here because I don't want to, I know this is just supposed to be a brief little overview, in kind of summary of, you know, what we've done for 20 years. Sure my slides are working. Oh man. All right. So, my group is largely focused on biocatalysis, you know, for various, you know, target or reasons. So some examples of recent projects here are, and I'll come back to a couple slides, using anaerobic alkane activation mechanisms and putting them into E. coli to make to get E. coli to eat up alkanes and make products from those. we do a lot of directed evolution, and as a result, we end up designing a bunch of genetic switches and molecular reporters to enable high throughput screening. And more recently, I've been collaborating a lot with Dr. J.C. Conrad in my department in engineering bacteria to express different appendages and things, and study how that affects interactions with surfaces and polymers and things like that. So I summarize most of my funding throughout my career has been NSF, and I threw together a bunch of numbers over here on the bottom right. So an overriding theme, my whole life has been directed evolution. And we early on, realized the need for developing high throughput screening systems. And we've been leaning a lot on the use of transcription factors. So, there's many ways of doing this. Here I'm showing also people like to use riboswitches, things that can respond to small molecules and then impact gene expression. We don't use those. We use bacterial repressors and transcriptional activators. And so the general, overriding kind of theme here is have a transcription factor that responds to a small molecule that can report on the presence of the small molecule. And if you have that, then you can do a lot of pathway engineering to make that small molecule. So a lot of times you don't have a natural existing transcription factor with maybe the specificity or sensitivity that you want towards your target compound. So then you have to first engineer that. So it's like a dual directed evolution process. Engineer the transcription factor to be a good reporter for you, and then use that to engineer the pathway that it's reporting on, or the enzyme, or whatever it might be. So here's just an example of the kind of workflow. It's nowadays, I think, pretty standard, but we've been doing this for a long time. This is for the case of engineering Ara-C, which is the natively responds to L-arabinose. It's a very popular inducer system for gene expression in E. coli. The promoter is P-bad. And so we've done a lot of work over the years with, figuring out how to make the right libraries, make big libraries, combine the use of facts and selections, and have kind of like a cool workflow to kind of at the end generate any number of Ara-C variants that respond instead of arabinose to other compounds. So, for example, just go from L to D. And so we've done over the years lots of things. We've done a lot of work with having E. coli, engineering E. coli to make this compound TAL, triacetic acid lactone. And we have all sorts of sensors with different specificities for that. Vanillin, salicylic acid, and others. I didn't put anything there for or selenic acid. So that's an example of engineering the transcription factor, just a very broad stroke example. And so here's an example of using it for pathway engineering. So earlier on, working in the Yan lab, engineered E. coli to oversynthesize salicylic acid by putting in this, multi-gene pathway and preventing, the escape of charismate. And so we wanted to see if we could then optimize this pathway. It's a bunch of genes, and it's not optimized. They're all just being expressed from synthetic, or maybe, I don't even remember which, what the promoters or RBSs were in the original, constructs. So we took our salicylic acid responsive Ara-C variant and first demonstrated, okay, these cells are only going to make GFP when the pathway is complete, and they're making salicylic acid. That all makes sense. And so we took the pathway, which has ultimately we randomized, or not quite randomized, we synthesized a series of ribosomal binding site sequences for 6 different genes being expressed in the pathway. So it wasn't completely combinatorial or random. It was somewhere around 39,000 combinations, and then use the sensor that you got to screen all those actually pretty quickly. the harder part back when we did this was synthesizing the RBS libraries and things like that. But we showed that this works, pretty. It's quite effective. It's harder actually to understand why the improved production is improved, because then you look at the transcription levels of the different intermediate genes, and it's like, this doesn't make sense. That's a whole nother, study. So more recently, we've been interested in this alkane activation in E. coli. So a big challenge was simply getting functional expression of this enzyme system. It runs with a glycyl radical enzyme, and it essentially adds fumarate. This enzyme adds fumarate to alkanes to generate methyl alkyl suctinates. That's why it's called a MAS. And then those can be further metabolized, not necessarily with ease, but ultimately processed through beta oxidation to make building blocks that can be used for metabolic engineering. And so our first, challenge was in getting this system to be functionally expressed in E. coli. And then we wanted to start engineering it to be able to activate smaller alkanes. And normally it works on about C6. So here's a compound that we can make now pretty readily in E. coli. It is a methyl pentyl succinate. So it's from the addition of fumarate to hexane, C6. And we could do C5, C4, but it'd be nice if we can get that all the way down, the holy grail. Can we turn an alkyl succinate synthase into a methyl succinate synthase? So here's, alkyl making alkyl succinate. What if we can do methane to make methyl succinate? Then boom, we have methane activation and conversion metabolically to other things anaerobically, which is different from requiring all the other things if you're trying to do things with oxidative systems. So we want to make a biosensor for methyl succinate. We found in the literature that there's this ITCR transcriptional regulator that responds to itaconic acid, which has a double bond here. It's methylene succinate. It doesn't respond to methyl succinate, which is different just by that difference in the double bond. So, okay, that's amazing in itself that double bond, it makes such a difference. But we did directed evolution, and so we've made an ITCR that responds now instead to methyl succinate, and so we can use it in working towards engineering the substrate specificity of the methyl the MAS enzyme. But that's a big jump. So we're actually first working towards just increasingly shorter chain alkanes. So here, the first step was converting our methyl succinate sensor to a methyl pentyl succinate sensor, because we can make a bunch of this guy. So we have a sensor for this, and that allows us to actually monitor the continuous synthesis of alkane activation in culture E. coli, that are expressing the MAS system. All right. And then finally, been working a lot with my collaborator, JC Conrad at UH, in doing really cool things that she understands a lot more than I do about how changing the expression levels of things like curli and fimbria and flagella and other surface appendages in different ways affects the way that E. coli interacts with emulsions, droplets, moves things around, or forms different types of affects the biogenesis when interacting with different types of polymers and surfaces. And so I've been working on things in that direction for quite a while now, and we're still kind of seeing where it's all going, but there's a lot of interest in the soft matter community on those types of things. 10 minutes, I will stop. And thank you again for the opportunity. And I'm happy to, or I'm looking forward to hearing Linna Ann's presentation.
Thank you so much. This is a wonderful. So I didn't tell you earlier, but your earlier work when I started my position as a professor actually influenced a lot regarding sensor development. So I'm really thank you for your pioneering work at the time. So I really appreciate it. And also your, work on tar actually also sort of, inspire us. So we are moving into the even tar making production right now. So, but we are very slow right now. So I don't know when we'll get the tar production soon. So anyway, fantastic. Thank you so much. This is a really amazing talk. Now the main speaker of today with a longer introduction. Professor Linna An received her bachelor's degree at University of Science and Technology of China in the Department of Chemistry. After getting her bachelor's degree, she moved on to get her PhD at the University of Illinois, Urbana-Champaign, or UIUC, with Professor Wilfred van der Doorn. Her PhD thesis focused on the discovery and engineering of natural product biosynthesis systems, particularly antibiotics. After her PhD training, she moved to University of Washington to work on computational protein design with Dr. Dave Baker, a recent Nobel laureate, in recognition of his computational protein design tool development, including Rosetta and other tools. Currently, Professor An is a tenure-track assistant professor at Rice University, Bioscience Department. She just started her faculty position, I think this year, and I want to congratulate her. So congrats again. Her lab at Rice University focuses on using and developing machine learning method for functional protein design with a focus of sensor and enzyme design. And I believe this is a very hot topic at this moment. I may need to call today a protein engineering day by using both approaches, of course, directed evolution and computation-based design. Or it may be a Houston day because both speakers' institute are located in Houston. Linna, thanks so much for your time today, and please take it away, all yours now.
Thank you. Let me share my slides. Okay, thank you all for the great opportunity and then for the great introduction. And then this is Linna, and then I'm going to use today's 30 minutes to roughly talk about my past research and then what my lab is going to do in the future years. So again, thank you for the exhaustive introduction. So I probably don't really need to spend time on my experience. So basically, I want to point out how I turned to computational during my whole experience. So I was really fascinated about biosynthetic systems and functional protein designs, especially protein interacting with ligand, etc., during my graduate research. So at that point, the major way for you to improve native protein, especially native enzymes, like what Patrick is doing, is you do something called genome mining. You look for homologs of the proteins, and then you do rounds of direct evolution on engineering different profiles to improve the protein into the direction that you want. So this method has been proven to be really successful in many cases, like terpene synthase cases and Patrick's cases, examples he showed. So, however, for many other proteins, it's just really complicated system. So direct evolution is extremely slow on actually improving the profile we are looking for. So I was thinking, okay, probably there are some other ways to make it faster. So that's how I was looking at computational method and entered the functional protein design lab at the David Baker lab at University of Washington. So that just to give people a quick overview on what's a problem is in the protein design field. So basically, for protein design, the first thing is you can do something called protein structure folding prediction. Meaning you can totally understand this as you are drawing a relationship between the gene sequence to its amino acid sequence to exactly what's the structure it's going to be folding. And then this relationship you are establishing is through building some sort of like a model, especially recently pioneered by machine learning and deep learning models. And then once you know how to predict, then you can actually do something what we call as design. So meaning you start with some sort of like protein functions that you are extremely interested in, and then you are trying to reverse the model of like prediction to generate the sequences which will in your hope like folding into the structure that you are interested in. So in my lab, I want to talk about like what's the position you can think about for protein design labs. So pretty often we talk to like the doctors, biologists, industry people, and then get the phenotype information they want. For example, this is including some reaction they want to catalyze, and then some protein they want to target, etc. And then we work with the mathematicians and computer science collaborators. They, of course, provide us some suggestion on improving the model architecture, etc. And then, like we adopt their methodologies to make what we call as generative model for protein generation. And then because we can control what type of protein, what's the size, what's the profile we want to generate from the protein. So our protein generator is really fitable for doing high throughput screening through an automated and also assay. And our goal is to deliver the pilot molecules as fast as possible to our collaborators in their complicated like real world systems. And the advantage is what we can have is we can also take the information we got from the experimental and then feedback to the model systems. So of course, like you can understand as the middle part, which 50% of the computational and 50% of the experimental work is majorly done by people like me who are leading a protein design lab. And of course, it's extremely honor to talk to all the audience here because, like, as you may notice, there is a lot of common sense between what we do and also what's the majority of the synthetic biologists are doing. And then there are definitely lots of crossovers and collaboration opportunities that I'm genuinely looking for. Okay, so now let's turn more deep into like what can be done in a protein design lab. Pretty often how we do is we start with a desired function, and then people like us make a hypothesis what type of molecular interaction can achieve these functions. So for computational tools, you are literally only using it to make the desired interaction achievable through a higher speed and efficiency, and then in the end you were able to obtain the protein sequences you think is going to be folding into the structure with the hypothesized function, so that you can actually obtain the desired function faster. So that's to say, it's actually really just a new method to explore a really large energy landscape more effectively. It's not really fundamentally that different except for efficiency-wise compared to traditional. For example, traditionally, you may have an assay that generates, let's say, 100 data points a day or a week for you to understand the whole energy landscape you are dealing with. Now, what we can do is having computer do really fast screening so that we can generate 10,000 data points per hour, something like that, to rule out the bad interactions possibilities and then focus on the real interactions land into the energy well in the whole energy landscape. So although this is philosophically pretty similar to traditional protein engineering, the efficiency change actually made a huge difference, which is making the previously unengineerable problems engineerable. Now let's pivot to what my lab is doing. So in my lab, like we majorly use and develop machine learning algorithm for functional protein design, and then we focus on two domains of the work. And then in the coming part of the talk, you will see why these two domains is actually like method-wise really acting with each other. So the first domain we work on is that small molecule like landscape monitoring. So basically sensors and small molecule mediated gates design. And also on the other hand, we also do biosynthetic enzyme design. Today, I will majorly talk about binding and then touch upon the sensing and a little bit about the enzyme design work. So if you want to know anything more about the details of the methods we develop, like please do feel free to reach out, and then everything we develop is definitely for sure like open source. Okay, so now let's look at the ligand binding problem. So the problem where the task we're trying to solve is like if you are starting with a small molecule structure and then using all different type of like computational operations, in the end, what we want to present to you is a protein which is designed from scratch with certain interaction ability. Again, all the protein I'm showing you in this talk are generated from computation method from scratch. Non-mutational and direct evolution method we use to mature the interaction ability. So once you have a protein which is interacting with the ligand, of course, then you can do lots of things, including binding, sensing, enzyme design, etc. So this is actually not really super easy problem to resolve. The major reason is, first of all, you have really large landscape to look at because you have to enumerate the ligand rotamer, the protein scaffold, set chain interactions. And also at the same time, this is actually not really a super trivial sampling problem. You also have to be extremely accurate. The major reason is because protein small molecule interface is relatively small, and there is strong evidence showing your protein backbone accuracy have to be at the level of roughly 0.5 angstrom to have the interaction between protein and small molecule really achievable, because those interactions majorly are short-range interactions such as polar interaction, pi-X interaction, etc. So it's actually a really hard sampling problem that we have to resolve here. So just to let you know what type of like achievement we were able to demonstrate for the past like years. So in the recent published paper, we were able to demonstrating like successfully binder design against like critical metabolites such as cholic acid, and binding against like central some larger ligands such as cyclic peptide, and also particularly interaction with like polar ligands such as methyltrypsin and siroxane. So because we have been able to achieve repeatedly success against generating binder against multiple ligands, so we are pretty confident our model method is mature enough to start to deal with more real world problems. So now let's just give you a brief introduction on how does the whole pipeline work from the more high level of observation. So first of all, you need to generate the ligand structure. At this point, we don't really do anything special with ligand structure generation. As long as this structure is physically allowed, it doesn't have to be in the low energy conformation. And then as long as it's reasonable, it can be used as an input. And then the next step is we use machine learning method to generate a protein scaffold, which is basically the protein backbone, C, alpha, and O positions around this ligand. The goal is to generate a protein with high shape complementary to the ligand so that it can be predicted with highest interaction probability between the protein and the ligand. So this problem in the protein design field is mostly referred to as conditional scaffold generation problem. And then I personally involved in like AlphaFold 2 based hallucination method development and also C-alpha diffusion method development, which I will quickly mention in the coming part of the slides. And then once you generate the protein scaffold around the ligand with relatively good shape complementary, then what you can do and with optional is you can enumerate the protein local structure and also the ligand docking structure to improve the protein and small molecule interaction. You can sort of imagine this as like you have a energy landscape and then you land into an energy well, and then now you're just nudging it so that you are improving the location of the well that you are in. So you sort of can treat this as a docking problem, and then there are methods developed for as energy-based GA ligand docking, and also some machine learning-based method developed for this, which I won't really talk about today in this talk. And then lastly, now you finally land into a scaffold, which is with a small molecule docking structure. Now, what you actually need to do is generate the sequence of the protein. This is actually the step where you're generating the particular interaction and sequence of the protein to achieve the docking conformations between the protein and the ligand. So this is pretty often referred to as fixed backbone sequence design problem. And then, like I have been involving in developing of the graph neural network ligand NPN for generating protein sequences with the providing of the protein small molecule ligand complex conformation. Pretty often we iterate between the last two steps to achieve the best sequence with the most optimal protein small molecule co-folding structure. The aim is, you can imagine this as we want to search for a convergence between the small molecule between the protein sequence and the small molecule protein docking conformations. So that is giving you the highest probability with the interaction. Okay, so now let's take a slightly more deep dive into the scaffold generation step. So back in 2021, we have a simple idea. So for protein structure generation, what we can do is, like, first of all, just starting with a small molecule ligand, and then use computer to generate all the proteins with like the pocket inside. And then in this way, we can just use quick docking to identify optimal protein scaffold around the or with the docking result to the ligand, so that then we can continue the protein small molecule interface design. So now the problem become into, like, how do we effectively scanning all the proteins with pocket inside around the sequence space we are looking at. So that's how we we build this AlphaFold2 based hallucination method. So the idea is, in the end, we want to land into a protein scaffold with pocket inside. We change the problem into generating pseudo-cyclic proteins, so that because it's pseudo-cyclic, so it's always going to have a pocket inside. How we do it is we initiate the protein with a repeated sequence, and then we pass it onto a protein structure predictor, so that we can generate the protein scaffold, rough scaffold. Based on this rough scaffold, we can extract its like folding likelihood score and also the geometry score. Combining these two scores, we write a loss, so that and then put that into a gradient descendant type of like algorithm, so that you can keep optimizing the loss, so that you in the end, like, directly optimizing the foldingability and also the scaffold of the protein. But what we found is, actually, as expected, within probably 10 to 20 steps, actually we can have pretty good convergence between the scaffold we generated. Afterwards, we redesign the sequence of the protein, so to improve the solubility likelihood to in the end generate the protein with pocket inside. So since this is a computational method, so you can do it like for 1 million times on computation without like you being in the wet lab. So that's exactly what we did. We obtained a lot of protein with all beta like structures, alpha beta structures, and then all alpha structures. So of course, the common point they have is they all have a pocket inside. So in the end, we obtain like almost 10,000 unique scaffolds clusters with different second structure and also different pocket property size. Of course, at that point, one thing we ask ourselves is, are we purely generating really beautiful models of the proteins, or we are actually generating some protein which is really folding in reality? So what we did is randomly picking 96 proteins and then express them in in small scale 1 mil culture, and then test them like on chromatography and challenge them to CD folding. So gladly, we found almost all the proteins are folded pretty well with monomeric dispersed peak on sex exclusion column and also the expected CD profile, suggesting their folding are with expected secondary structure. At the same time, we were able to get a few crystal structures demonstrating our design protein are with expected high backbone resolution, and then also with a pocket inside. So afterwards, we put the ligand in and then do sequence design as we referred to, and then in the end, they can become binders. Parallelly, like around 2022, we realized that first of all generating pocket and then put the ligand inside for protein design, for small molecule protein interface design is good. However, we want to make the whole procedure even more effective. At the same time, the vision, the image generation field actually demonstrated is a pretty effective generative architecture, which is a diffusion-based architecture. Basically, what you can say is that if you put lots of image data into this diffusion model with keeping adding noise, in the end, like having a well-distributed noise distribution, you can in this way train the model to build the relationship between the image and also in the end the noise distribution. And then actually, what people found out what you can do is that you can do the reverse to initialize with the noise distribution, and then slowly, stepwise remove the noise, and then in the end, you can actually generate a image with pretty high readability. So if you played around with like image generation engines such as DALL-E, those are actually built based on this type of algorithm. So what we found out is we can actually do similar things with protein scaffold generation. We can start training with a PDB database, and then in the end have the distribution of the noise. And then when we generate, we can initialize with the distribution of noise, and then slowly remove the noise to generate the protein structure, which is pretty much fitting the distribution of the PDB structure profiles. So based on the algorithm developed by Dr. Joe Watson, working with David Jurgens and two other students at the Baker Lab, we were able to expand this method to be ligand-aware like diffusion method. So now, what do you mean? What we have been achieving is like, you don't really have to generate the protein from first and then put the ligand later. You can start with the ligand and then generate the protein around this ligand, and so that you directly get a protein scaffold with high shape complementary around this ligand. Okay, so now you can use at least two major different methods to obtain like design ligand binding proteins. So one of the questions is, how do we actually validate them in experimentally? What we found is like for some more challenging ligands, such as polar and flexible ligand, the success rate is around 0.5%. So we still do the high throughput screening to test like a few hundred to a few thousand designs at the same time. What we do is we put the gene which is encoding the design protein into yeast, and then they can be expressed on the surface of the yeast with a certain C-terminal c-MYC tag. So in this way, we can add the fluorescent labeled small molecule target into the yeast, and then use antibody to stain to show the expression of the protein. So within probably 2 weeks, we will be able to build the yeast library, which is expressing the design protein, and then we can take a look at using high throughput cell sorters to look for double positive signals and collect this population. And then we use like next generation sequencing all calling PCR to identify the best hit among our design binders. And lastly, we always do a step of like purification to verify the binding between the design protein and also the design target. Of course, to some of the target we found out if they are rigid and hydrophobic, actually we can achieve pretty good success rate around 5%. to 10%. Meaning, at that point we don't really try to do a library. Instead, we directly order a plate of gene or half a plate of gene, and then pretty quickly we can do Golden Gate type style of assembling for plasmid generation, and then express them in small culture of E. coli, and then through quick purification and high throughput and mid throughput of the interaction measurement using fluorescence polarization and surface plasmon resonance, we can also quickly to identify early pilot binders within 2 to 3 weeks. And now let me just show you some of the binders we got. For example, we were able to directly get a nanomolar binder against a metabolite such as cholic acid, micromolar binder against methyl and nanomolar binder towards polar metabolite ligand such as thyroxine. More recently, the student that lead was able to target 3 different ligands at the same time. As you may notice, these 3 ligands have exactly the same small molecule scaffold, only with different decorations at different ring size. So so they are actually really close, resemble small molecule analogs. So he didn't really put too much thought into how to differentiation design. Instead, he just purely start to design against all of the 3 dyes at the same time. And then directly he was able to get nanomolar and sub-nanomolar binders with high specificity against different individual targets. Of course, then you may be interested in how specific our de novo design binder are in cells or in more complicated situation. At this point, of course, we have more limited amount of data. However, recently we have some really strong data showing all the designs remain highly specific to their design targets in cells. So again, for the dye binders we talked about in the last slides. So all those 3 structures of the ligand also have different fluorescence properties, so that we can actually perform high resolution, super resolution imaging using those 3 dyes. So what we did is we cistronic express all the 3 design proteins, tagging to different cellular organelles such as mitochondria, nuclear, and endosome. And then we add 3 dyes simultaneously with 2 nanomolar concentration. And then after 5 minutes of incubation, we perform 3 times wash, and then directly image the cell with fixation condition, all live cells condition. So many super resolution imaging conditions, we were able to find many cells with simultaneous transcription of all 3 dye binders. And then because with incubation of all 3 dyes, we can say the dyes specifically light up the region that the dye binders are localized to, suggesting the dye binders remain specific against their design dye. And then this also allows us to perform multicolor, multiplex cellular imaging on the different conditions. So now I want to also spend probably in the next coming 5 minutes talking about the progressing in sensing and also enzyme works we do in our lab. So I have always had the invasion of I hope to build a toolkit so that allow us to do in vivo, in vitro, and cellular imaging to light up the ligand of interest. So that for the biochemical and therapeutic applications. This is a particular technique that is available for protein imaging, for transcriptomics imaging, for example, work developed by Xiaowei Zhuang lab, etc. However, just really lacking and not there for small molecule and peptide imaging in cell and in vitro. So to achieve that, what we did is for all the binders we develop, actually we purposely make them into pretty simple scaffold, so that when we remove the loops between the subdomains, you can actually turn that into a ligand-induced oligomerization protein. Then by fusing them into modular sensing signaling modules such as nanopore sensor or split GFP, etc., we can actually achieve like ligand-specific sensing modalities. On the other hand, we can also make the protein larger so that they are easier to engineer and then turn them into ligand-induced conformation changing proteins or ligand-induced dimerization proteins. In both cases, we can like fuse them to split fluorescent proteins of split like luciferase, etc., and then achieve ligand-induced signaling for like in vitro and in vivo sensing. To just show you some data, for example, for the nanopore sensor, for nanopore, what happened is you build a channel around the membrane or solid base, so that like normally when the analyte is not really in the solution, you can achieve a stable like baseline that is showing like empty the signal. However, when you have the ligand inside, as expected, the pore get blocked so that you directly see a disappearance of the signal, which is indicating like we were able to build like pretty modular ligand specific sensors using the nanopore like sensing modality. And lastly, I want to quickly touch about the enzyme design. Sorry, I know I'm slightly over the time. And then for once you have the small molecule binder design like method, you can actually pretty easily turn that into a enzyme design pipeline by instead of binding substrate or product, you focus on recapitulating the transition state interaction conformation for a reaction to be achieved. So traditionally, since I have been in the enzyme engineering field for my graduate school, I realized like the limitation of the enzyme engineering is literally just one, if you don't really have a sensor that is presented, such as in Patrick's work, you really suffer from low throughput engineering, which is just really difficult for you to for you to do for enzyme activity improvement, because the potential space you are facing against is really huge. However, most of the time, if you have only chromatography based activity assay, then you're only having like the probably 10 to 20 mutations to deal with. That's just like not really enough for you to improve the enzyme activity. On the other hand, many enzymes are just too slow to evolve because the nature of their reaction is just too complicated. So what we envision is that we can build sensors from scratch based on the product or the substrate of the reaction, and then put the sensor into the microbe together with the enzyme. So in this way, we can in situ report the enzyme activity of stationary phase using high throughput methods such as cell sorting. And then by this method, we will be able to build large sequence activity relationship data set in a relatively quick fashion, and then build small machine learning models using methods such as active learning to generate new predictors for mutants, so that like potentially we can accelerate the enzyme activity improvement schedule. Just some preliminary data. My graduate student Hayden Stagel, he was able to focus on one of the cancer-related natural product, and then computationally build like binder against them with really good like prediction fidelity. Especially, we also use like traditional computational method to predict the binding energy between our design and also the native binder. Suggesting our design can be achieved in a pretty high confidence with energy-based like method and also machine learning method. Currently, we're in the process of testing these binders, hopefully, and then turn them into like sensors. And then on the other hand, besides just making like reaction sensors, we also aim for doing first principle based enzyme design. So basically, the idea is instead of actually following traditional pathway, which is discovering enzymes from nature, and then engineer them for our need, and then individually engineer for their different properties such as solubility, activity, and specificity, etc. We directly starting from the theory and the predictive mechanism, and then think about all the conditions we need to consider during enzyme engineering, and directly design enzyme based on our needs. So this is basically using organic synthesis method, a principle to really tackle like biochemistry method, which will really provide an alternative method for biochemistry study. And then, so just, of course, people may understanding enzyme design is just really, really challenging. However, I want to tell people is that for the past 2 years, there's really a lot of progress in this field. For example, literally just in past year, there were 4 different papers put out from different groups, really describing the recapitulation of hydrolysis reactions. That's actually not a super easy reaction because it's technically a multi-step reaction, which requires you to first form a tetrahedral intermediate and then through different methods to kick off this intermediate through a hydrolysis, pretty often water-mediated like hydrolysis reaction. So using different machine learning methods, groups from like different continents of the world, we are able to directly generate like hydrolysis using different mechanisms such as serine hydrolysis, cysteine hydrolysis, and metallohydrolysis. The key point is everyone found out if you have a really, really good method to recapitulate the reaction, catalytic triad and catalytic center, then pretty high chance you will be able to build this like active enzyme with actually pretty different like protein scaffold. So in my lab, we focus on biosynthetic enzymes, and then to also take advantage of existing crystal structure and computational chemistry calculation results, and then directly generate a new protein catalyzing the reaction that we draw by the mechanism, and then from scratch. So hopefully we can in the next coming years to tell you about our progress in directly generating these like biosynthetic enzymes. And then one more thing before I finish my talk. So actually, like we realize this field really needs the participation from different people from different fields, including biochemistry, machine learning, CS, and biologists and doctors in different like hospitals. So we are building this machine learning for biochemistry club. Through hosting seminars like what Tesla is making here, and also and then we also aim to like organizing like local happy hour around the Houston areas to bring people together and then brainstorming ideas together. So at this point, we already be able to schedule 3 talks from like really high level labs and like industry partners. So if you are interested in getting involved, please do scan the QR code and then just like getting onto our email list and then follow us on the social media. With that, I'd like to thank the audience for staying with us like for the past like probably 40 minutes. Sorry, we definitely go over the talks, and then do let me know if you have any questions. Thank you.
Fantastic talk. I have some question, but before I'm asking, I want to ask Patrick still there, or Patrick need to go to the airport?
I'm here.
Okay, so do you have any comment or question to her?
Are you a part of these dinners that we do, that Ross keeps inviting me to once a month?
Yes.
Oh, have you gone? I haven't gone. I should go. I keep saying I will go.
You should come. You should come. It's really fun. I have been always going. I'll be honest. One of the reasons is my partner haven't really moved here yet. So right now I'm literally living a bachelor life.
Oh, I gotcha.
But once my partner come, like he will definitely be happy to join as well. You should for sure join. And then many of these events are at like family friendly dinner. Yeah. I don't have kids yet. However, like other people have bringing their kids. I feel they had fun as well. So you should definitely come.
Okay, I'll do it.
So
that's my only question for now. Thank you. Do
you have any other question or comment?
No, just I mean that this is all extremely impressive. I'm aware of some of this, and I think you're in a great place at Rice. I think they got lucky to get you, and I look forward to seeing what you do there, and good luck and congrats.
Thank you.
so much. Thank you so much, especially you are traveling and a safe trip back to Houston, I believe.
Yeah.
So,
yeah. Thank you.
Thank you. So let me ask one question. So this is impressive. Of course, I have been using the AI, machine learning, and also AlphaFold, Rosetta, for many years, and that makes my life much easier. I didn't like the protein engineering when I was a graduate student because I need to screen like 30,000 different colonies by hand
Oh
without
my
any
God.
machine. I was robot. So after that, I didn't want to do any protein engineering until Rosetta AI, those things, come to my mind. That's 10 years ago. So question I still have is you show at the end the enzyme engineering. That's pretty impressive. But what I did during, my PhD degree a little bit 20 years ago, still very challenging because even the computation method we have right now should kind of have the real prediction how transition state protein structure with the chemical reaction kind of interact. After that, as you mentioned, we need to talk about real reaction, not just binding. So could you kind of comment on the challenges we still have? Because this is not easy problem. To me, I mean, the sensor development using AI much easier because that's only probably talking about the binding.
Yeah.
But it's a completely different story in terms of the enzyme.
Yeah, I totally agree with you. And then, so this is why I'm really trying, when I'm talking about the enzyme direction, I'm really trying to be more softer during my talk. So because the reason is because first of all, so at this point, I fully understand for the transition state, for most of the reaction, we don't know. For some of the reaction, we have a good picture. However, we don't really know how accurate we are. So let's say like, when I talk about like people are, multiple labs were able to design hydrolase. There is a reason why we target hydrolase as our first candidate because hydrolase is almost one of the reaction which is slightly more complicated, and then we just have so much understanding on how it works. So I currently like I think there's a consensus is that currently the models can handle binding to some extent with, I would say, pretty good accuracy. However, it doesn't really deal with dynamics. It doesn't really understand too much about like different like conformation changes. So first of all, you definitely have to pick a right reaction that only have like really one transition state you have to tackle with. And this transition state, you just have really high understanding on what is going on. So it's not like we can design any type of enzyme at this point. So, however, this is also a new field. So there are plenty of like well understood reactions we try to focus on. And then on the other hand, like not my work or not the things I'm going to do in the next few years. However, there is already a strong progress on like using machine learning to accelerate DFT calculation and MD calculations. Example, recently there are European groups in Germany published like the what is called maze architecture. I think it's just really impressive. So meaning like if the models can at least accelerating the local DFT calculations. For example, for all the enzymes I design, if I cannot really check with like with MD real MD individually, however, I can check with really quick like machine learning accelerated MD or DFT calculation in relatively okay resolution. That's still going to be significantly helping the success rate of enzyme design. And then lastly, most important, you mentioned, okay, so we are still using the binder design strategy to approach enzyme. That's really important, meaning like when you are doing the experimental validation is really important because there is definitely a gap between what you are dealing with computationally and experimentally. So experimentally, you should never screening for let's say intermediate like mimic binding, you should really screening for the actual reaction. So that's to say like so we're probably still far away from we just design two things and then one of the things is active. We still really want to build up the force of automated like screening. If we can generate a lot of data points, we definitely also improve our model as well. That's to say, like at this point, like programming in my lab together with like Rice AI Force, we're also putting a lot of effort in actually recruiting high throughput like structural biologists and also like automated protein assay platform, etc. We're definitely like recognizing AI is just like helping to make things faster. It's not really the solution yet. We do hope like for example, after 5 years or 10 years, our success rate is high enough that you only need to test like 10 things and then get something starting.
Sure, absolutely. So, even 20 years ago, when I talking about my PhD work, and at that time I was so excited by seeing the de novo enzyme design combined with the directed evolution, start to see some enzyme, but after 20 years later, we see, tremendous achievement. So I'm very, optimistic. And then now I believe I spoil my people, my own lab people, because by buying the liquid handlers and the automation tool, but that kind of speed up or the entire, progress, I believe. One
Yeah,
last
I agree.
question. Yeah, because I need to, close, but one last quick question. Uh, so you show amazing binders that is, to me, mostly hydrophobic, I believe. And then that has more interaction between protein and the small ligand, but still that's a big enough to me. But I wanted to kind of see much generalizable kind of the AI designed protein, including small molecule like, I don't know, succinate, acetic acid, even that small. So
Yeah.
what is the real challenge going that direction? And also I heard, also based on my experience, some protein, structure like, I don't know which one is which one, but globular protein or, helix protein, some of them is working very well, but the other is not working well. So can you kind of give some insight, why is the case and how we overcome?
So for small molecule binding, like of course the thing what we show so far is like we definitely show diverse enough to some extent, but not really to everything. For example, all these ligand, like I would say, the one with highest like flexibility freedom is probably these two. Yeah. Then so the major issue for like really tiny ones, for example, acetic acid and then alcohol, things like that, is just really, really small so that it's difficult to initialize like the initial interaction because so if the method is good enough to be extremely accurate from the first hand, it will definitely make those type of like interaction build up like way better. Another direction which the whole field is actually working towards is like affinity prediction. So for really, really small like ligand, even if your model is highly accurate, like building all the short range interactions immediately, like if the affinity is not really good enough, you wouldn't be able to see that as well. So unlike for like, let's say for slightly larger ligand, for example, for methyltricide, it's definitely more polar and flexible compared to other ones. And then the interaction building is complicated and more challenging. However, as long as you are really accurate, there is a really high chance you get a binder. However, for really small ones, like let's say acetic acid, even if you form hydrogen bonding against all the polar atoms, like the hydrogen bonding strength really matters because you have a huge dissolution energy to fight against. So even if you really form those, like if the affinity is actually, let's say, millimolar or high millimolar range, you wouldn't be able to see that in experimental as well. So that's big challenge. So I think like currently the whole field is moving towards the direction of one, improving improving accuracy even more. Two is figure out how do we predict affinity, or at least ranking the designs with affinity. I think if those two points get addressed, then the really small and challenging molecules, especially for example glucose, will really be resolved for binding problems.
I see. How about the globular protein versus helix protein? Which one is easier?
I think both of them is okay, because I would say currently that as long as it's a protein folding problem, it's not really going to be too crazy.
I see.
Yeah.
Okay,
So
let me check. Oh, go ahead.
I have a question.
Sure.
So I mean, definitely enzyme is more challenging than just binding, but for most of the scenarios where binding is of interest, it's accompanied by a significant conformational change, and there's lots of good binding that is instead inhibiting or not corresponding to conformational changes, as a result of the binding. So how do you, I guess, take that into account? This has been an issue, with my computational, collaborators for forever. You can design a good binder, but it doesn't mean it's going to actually do the next step.
Mm-hmm.
And I'm thinking of it from a standpoint of like a transcription factor, which, a lot happens after binding.
yeah, I agree. So that's to say like the approach. So there are two technical ways we are approaching this. One is to just have the model to be more flexible with understanding different conformations. So currently the model is more effective on just figuring out one conformation. And then currently we're developing models to be trained with multiple conformations. So once those type of model get like better performance, then this problem should be getting better addressed. And then on the other hand, I think transcription factor, actually like I'm just talking to Ross this morning, and then we have some program trying to be we are trying to figure out some partnership. So I think for transcription factor, another of the issue is like it's not a simple like protein small molecule binding problem which is triggering something. It's sort of a cascade problem based on my understanding of the model. So that's to say like in the end after some more time, like maybe the model can be structure wise capture everything. However, at this point, I think the most effective model to develop is actually still just like building the sequence to phenotype of the model to just like have a better efficiency on proposing better mutation and combos, because that's at least can be developed at this point now, and then just like show effectiveness within probably next 6 months. Because I'll be honest, I think like developing a model which is like understanding lots of conformation changes, cascading effect at the same time I would say it's definitely still like technical wise still challenging at this point.
Yeah. Maybe, I mean, have people looked into even just like saying, instead of trying to design for the best binder, maybe you look at categories of like maybe not quite the best. Maybe you look at the range of solutions where they're intermediate binders, and maybe there are more solutions that are also accompanied by the phenotype that you're interested in. Do you know what I'm saying? Instead of getting the best ones,
Yeah, so for all the enzyme design we do right now is like we only look at the reactions where the mechanism is super clear, and then there is a really strong evidence on we know with pretty high confidence what's the only important transition state look like. And then so what we actually pretty often found is like for those type of reaction is, for example, certain hydrolase reaction, and also the reaction we are making right now. they have a pretty strong evidence of like, because there is only one key transition state. So the set chain majorly, especially catalytic residue, etc., they are basically like adopting that pre-catalytic conformation for a pretty long time. So that's to say like if the goal is to really recapitulating those K residues at really high confidence with those conformations, you actually have a pretty high chance to achieve those reactions. And then that's something current model can do.
Mhm.
Yeah, so at least that's the principle how we choose why we focus on this reaction, not the other reaction.
Right. Yeah.
Yeah.
Mhm.
Okay, fantastic. So I probably need to say, even if a lot of people worry about AIs, whatever consequence, but still, experimentalist humans still necessary to contribute with the AI's help, I guess. So that's wonderful. But I excited to see, more, development in the AI and the protein de novo design, and that is probably something I wanted to see even 20 years ago. Okay, so let's close. So thank you all for joining and staying today. We'll meet again on April 9, Thursday, the same time, the same Zoom link. We'll have the Professor Julian Nguyen at UNC Chapel Hill and Dr. Ho-Sung Bae at UC Irvine. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again. And I stop recording. Just 1 second. Stop.